think I introduced myself on Monday, but I'll do it again. I'm Paolo Casale from Helmholtz Munich. That's the first time here at CGSI, and I'd like to thank the organizers for having me. It's been great so far. I look forward to the week to come as well with the longer program. So I thought that since it's the first time here, I would start by telling you a little bit about what we do in my group at Helmholtz Munich. So we focus a lot on genetic analysis, and this is a very general slide on the central dogma from genetics to phenotype through expression and proteins. And I just want to motivate why we do focus on genetics so much. Genetics is a special layer, is inherited at birth, and it stays constant in space and time, doesn't change, it's the same across all the cells in our body, especially if we think about germline genetics. While RNA and protein levels, they do change. They change with across cells, they change with tissues, with age, across disease states. And so changes in protein and RNA levels may be a consequence and not the cause of disease. It's just observational correlation. While if we do find an association between a genetic variant and disease risk or a phenotype, it is likely that this is pointing towards something that is upstream, the manifestation of a disease. And this principle has been beautifully shown now 10 years ago by this paper, which was published in Nature Biotech by Jia C, where they showed that the proportion of drugs with human genetic support increases significantly across the drug development pipeline. This means that successful drugs that tend to have support from human genetics. And people have known this for a very long time. We've been doing genome-wide association studies now for 20 years. And we now know of thousands of genetic loci associated with human disease. However, for most of them, we don't know what they do. We don't know what's the functional mechanism. We don't know the cell type, the tissue. But nowadays, we have access to many different intermediate phenotypes that we measure alongside with genetic data in large biobanks. And these range from genomics data, such as single cell, to medical imaging and clinical readouts. And of course, thanks to this large-scale data, we have new opportunities. We can study the effects of genetic variants that we know being associated to disease in cell and tissue phenotypes and understand what they do. We can identify new disease biomarkers. We can maybe also try to learn something about disease subtypes. Of course, these opportunities come along with many challenges, mostly computational challenges, and that's what, with my team, we work on at Helmholtz Munich. We work across different data modalities, and on Tuesday, I'm going to talk about mixed model for biobank scale datasets, focusing on medical records and single-cell RNA-seq. And today, instead, I'm talking about computational pathology, and specifically about an analysis that I did before going to Helmholtz Munich. I was working for a company in Citro here in the Bay Area until end of 2021. This is all published data. And in this analysis project, this was a collaboration with the Gilead Sciences. We analyzed 5 NASH clinical trials on patients with liver fibrosis to identify targets of NASH progression. And a bit about the data, NASH is typically diagnosed from a tissue image of liver biopsy, and we have 4,000 of these, and we focus our analysis on 2 phase 3 clinical trials for which we had about 1,600 patients, and for those we had genomics information, including genetics and liver RNA-seq expression. And these phase 3 trials, they evaluated the ASK1 inhibitor, and they failed at meeting the primary endpoint, so they were interrupted due to lack of efficacy. And so what we ended up with is a quite homogeneous cohort of patients across different arms of the clinical trials, together with genomic information, and there was a great opportunity to identify targets of liver inflammation progression. So a bit of an agenda. So I'll start by providing a medical background, and then the talk is going to be on 2 parts. First, I'll describe a supervised convolutional neural network approach to empower genomic analysis of histological disease traits. And then I'll extend some of these principles to go beyond disease labels, talking about how you can visualize tissue slides using unsupervised learning and enable downstream genomic analysis of imaging data. And the reason why I decided to go for this sort of analysis, a very applied talk, I thought this could be a motivating example to discuss some machine learning concepts that may be interesting to this audience, and I'll open a few parentheses to discuss these methodological details, specifically attention-based multiple instance learning, which is a type of supervised learning, and also we'll tell you about a contrastive framework for self-supervised learning working with images. Great, so let's start with a bit of medical background. So the non-alcoholic steatohepatitis, also known as NASH, is a multi-stage disease. About 25% of the worldwide population has simple steatosis, which is accumulation of fat in the liver, and this is a benign condition. Then about a fourth of the people having simple steatosis progress to non-alcoholic steatohepatitis, which is characterized by inflammation, hepatic death, and fibrosis in the later stages. And when the liver starts suffering because of these conditions, then the first symptoms include weakness, jaundice, itchy skin, and nausea, and the continued liver damage and progression of fibrosis ultimately may lead to hepatic cirrhosis with consequent liver failure and cancer, hepatocellular carcinoma. So the way we understand the progression of NASH and we assess the progression of NASH is through the assessment of an H&E-stained liver biopsy image. So take a small biopsy from the liver, and then this gets stained with H&E. This is a widely used technique for visualizing cell and tissue structure, and you may have heard about this modality for diagnosis cancer. That's also useful for liver diseases. And recent developments in AI and machine learning are enhancing the diagnosis from this and the preprocessing of these data sets. Then that's clear histological phenotypes showing up at each stage of this liver inflammation disease. So a bit more about the clinical endpoint in the trial we were looking at. The activity of NASH and the severity of fibrosis is assessed by pathologists by monitoring 4 different processes in the slide. Steatosis, which is the accumulation of fat in the liver. You can see these white balls, these are balls of fat. Lobular inflammation, which is the infiltration of immune cells in the lobule of the liver. Hepatocyte ballooning, which is a form of hepatic death, these faint hepatocytes that are apoptotic and they're dying. And fibrosis, which is the excess of connective tissue that results from the damage in the tissue. And as you can see here on the right, these are the scores that are used for assessing the severity of these processes, and these scores are very coarse. They are not designed for quantitative analysis. They're qualitative, and specifically the pathologists pathologists use these scores for finally assessing how strong is the state of NAFLD, of NASH, and how strong is fibrosis. They consider the sum of steatosis, lobular inflammation, and ballooning. And so, as I was telling you before, we had this opportunity that maybe with this trial, we can identify molecular features that are associated with the progression of these histological traits and maybe learning something about new potential targets. But the challenge is that these pathology scores are not quantitative. They're not designed for clinical analysis, and they provide a quite low resolution categorization of disease. So we thought about applying deep learning to enhance the resolution of this data set. So we aimed at predicting the pathology scores from H&E-stained liver biopsy imaging data, and by doing that, we could learn a continuous version of the scores. We then used these continuous scores for association testing with genetic and genomic information. So a bit of the methodology. So these H&E-stained whole slide images, they are huge images. They are composed of billion pixels. And so one common way of dealing with these images is to break them down into patches, into smaller patches, and focusing only on patches that contain enough tissue. Then these patches what we did, we used a convolutional neural network where each of these were fed to this network to extract features. And then we use an attention-based pooling mechanism to collapse these tile-level embeddings into a biopsy-level embedding for the whole slide. And finally, this biopsy-level embedding with a few layers were used to predict the pathology scores, which are scores at the slide level. So this type of setting is known as multi-instance learning, because we have multiple instances of a bag, which would be, the bag would be this whole slide, and the instances of the patches of the slide, and the labels that we have that we want to predict are at the bag level. So an important detail here, methodological detail, is this attention-based pooling mechanism. So if now consider a single score, for example, steatosis. What this attention mechanism does collapse all these tile embeddings in a permutation invariant manner to predict a steatosis scores. And now if we zoom in and we show a bit of the architecture, that's a specific implementation of the method that was published in ICML by Ilse et al. So we use a simple architecture from the tile embeddings to get importance weights for each tile, describing how important was each tile for scoring steatosis, but also a tile-level steatosis scores. And then the overall biopsy score was obtained as a weighted average of the tile-level steatosis scores using these weights. And importantly, as activation function of this layer, we use a softmax, which is quite standard because we want these weights to sum up to 1. And we use different heads, attention heads, for different pathology scores, because we wanted to give the model the flexibility of focusing on some of the tiles when scoring steatosis and on some other tiles when scoring other processes. So we trained these on about 4,000 slides and evaluated on 460 slides from different patients and clinical centers. And these are the predictions. We can see at the y-axis we have the predictions from the model versus the original pathology scores out of sample. And we measured the agreement between the predicted and the original scores by using Spearman correlation. And these numbers, they seem low, but they're in line with the cross-pathologist agreement. We checked that the model can capture different sets of tiles for different scores, and that was the case. And then if we, thanks to the importance weights that we learned, we can also go back and check what the model was focusing on when making these slide level predictions. And if we looked at the top tiles that were used for scoring steatosis, fibrosis, ballooning, and inflammation, we find very different features being picked up, which is what we wanted. And we also find that these are basically textbook images for these processes in a pathologist book. So we were pleased with these. Then to evaluate how well these continuous scores capture system-level traits, we correlated this with blood test biomarkers of liver dysfunction and fibrosis. And we used specifically a linear model from the scores to these blood biomarkers, either providing as inputs the original pathology scores or providing as inputs the continuous scores. And as you can see, we could always predict better these biomarkers, and for some of them, we saw a significant improvement. Then the part that we are most interested in, which is a genomic analysis of disease progression. So for doing that, we used a simple linear model for association testing, where we model as outcome the disease score at follow-up. So in the trial, we had the biopsies were taken at 2 different time points, at baseline and after 48 weeks. And so we model the disease score at 48 weeks and conditioning on sex, age, treatment arms, and powers of disease state at baseline. By conditioning of disease state at baseline, we want to capture progression in this association test. And then we test for association with genomic features. That's a simple log-likelihood ratio test, and we did 2 different analyses with associated with a baseline expression. So we assessed which genes had expression at baseline that was associated with progression. And then we did a genome-wide association study focusing on genetic features. And for the association with expression, we identified 37 genes associated with progression. What we saw is that if we instead consider the pathology scores instead of this continuous score to define disease progression, you find a much smaller subset. You only find 3 genes. So thanks to this really show the benefits of having this higher resolution in association studies that comes from this machine learning approach. And yeah, if we look at the biology of the progression-associated genes, we find lots of known biology and some new interesting pathways coming up, and same for genes associated with regression of fibrosis. Similar story for genome-wide association studies. So we used, in this case, a multivariate GWAS across the 4 traits I was telling you. We found 2 genome-wide loci. Bit underpowered, sample size is pretty low here, but then thanks to a series of post-GWAS analysis, we could learn about, we could collapse p-values into gene-level p-values and pathway-level p-values and learn more about the biology of progression. And again, when we used the original scores, we found no significant locus associated with the pathology scores. Okay, so this concludes the first part. I introduced a convolutional neural network that could predict NASH histological traits with cross-pathologies agreement, only using biopsy-level scores. And to do that, I introduced an attention-based mechanism that enable the model to focus on specific parts of the biopsy depending on the score. And finally, they showed that these scores that we derive, these continuous scores, they contain more information, and this was shown consistently. We're associating with systemic biomarkers, such as blood biomarkers, but also genomic features, including genetics. I'll stop here to take questions on the first part, and if there are questions, I'm going to go to the second part.
In your linear model, that G, can you explain what that is?
So this is the genomic feature we are testing. So it depends on the analysis we do. So for association with expression, this would be baseline gene expression. It's a quantitative measure of gene expression for both RNA-seq.
But lots of genes?
A single gene. So we test one by one. Okay. Yes. Okay. And then in the genetic analysis, this is instead a genotype vector. So these are number of minor alleles at a position at a SNP, and we do it for all genome-wide SNPs. So I guess your question may concern whether we model spatial distribution of phenotypes directly, or so the attention.
So it would be similar to mixed pathway in that sense. If it's not directly one tile independent in being of importance, but in certain cases, together with other tiles, it is of importance.
Yes, we don't model this in this framework, in this attention. But yeah, there are extensions of these simple attention models, for example, using transformers that can model indeed dependencies between tiles. Yeah. But this work is from before these transformer models came out for histopathology images. But it's a good point, yes. We assume independence, and of course, it doesn't have to be.
Could you explain in more detail what the H's are? What it's a tile. I'm not sure your vocabulary here.
Yes, this is the whole slide image. It contains billions of pixels, and so one can use a predefined grid to define subparts of the image. These are typically the size of them, 192 micron.
Well, how does a tile differ from a pixel?
It's bigger. It contains multiple pixels. Typically, it's a smaller image of tissue. Yeah. And in principle, one could go lower, super lower scale to single pixel. That probably wouldn't work very well. But yes, a tile would be a collection of pixels, and ultimately, this image is in our case, is a 256 by 256 image. So,
okay.
In terms of pixels.
And what are you measuring? Just the light?
This is RGB. So yeah, H&E-stained. The dyes are visible in the RGB, and they capture cells and tissue structure.
So you have 3 measurements per pixel.
Correct.
And then you
Yes, it's a standard image. Yes, correct. Standard image, and we use a convolutional neural network, right? As with
what
standard.
are you trying to predict? Agreement with the score of the expert?
Yes, exactly. Those 4 scores there, the pathology scores, that capture the 4 processes that are monitored when assessing the severity of the disease.
Where did you get this from? Like the latest?
These are from a central pathologist, that was involved in the trial. Actually, across the multiple trials, was the same pathologist assessed the slides.
So this is legal biopsy proof, right?
This is liver biopsy, correct?
And so how do you deal with edges? Because when you tile up the picture, you probably have lots of tiles with edges, right? When you are at the edge of tissue, probably you may lose a lot of information because it's a tile which is nearly 2 millimeters square, right?
Correct. So in this analysis, we did quite naively in a sense. So we just considered tiles that contained at least 80% tissues. That's the way we did it. Yeah. So we threw some data.
How did you choose the size of the tile?
It's a good question. So we tried with different resolutions and then evaluated the performance on a validation set before then applying finally on the test set.
So it's a supervised method, right? Like you are training based on the
Exactly. This is fully supervised. It's trained end to end.
pathologies.
Based on the pathology scores. The main challenge of this type of problem is that you have a huge image and only these 4 scores to supervise the feature extraction. I think that's the main challenge. And the way one can mitigate that is by using this multi-instance learning approach when one breaks the image into tiles and then feed each tile in a neural net as a single sample and then aggregate these tile level features that are extracted by the network.
And then once you have your higher resolution scores that you are obtained through this method, what's the variability of that? Have you assessed?
Good question. Not very high. Also, given the sample size that we have here, heritability estimation is particularly challenging. We got big standard errors.
Is it possible if you could do some analysis where it would be a clustering of mixed modeling, so you could get some types of the liver diseases, so some type groups where people have the same areas of bubbles. I'm not working with liver, but it's all those.
So, I guess.
Disease progression in the areas and then cluster them together.
So I'm not sure whether this is your question, but maybe one interpretation of your question is, can we do an unsupervised analysis of this embedding and do clustering, for example? And do we see structure that correlates with these features?
To see if there's differences in the people, in the 550 people, in terms of how if there's similar cases.
Yeah, so if we're talking about people and disease progression at the level of individuals, there's a lot of heterogeneity in these phenotypes, and unsupervised analysis would not be able to capture disease progression.
Oh no, it's not disease progression. It's just if there's similar cases in the group. like subtypes of the liver diseases?
Not obvious. So let's say the unsupervised analysis of this data did not show obvious subgroups. Great. I'll move on to the second part. So so far we've considered this is labels, and we use a supervised approach that could extract features that were specific to these labels. But the question we want to make in this part is, can we learn quantitative features of liver histology beyond disease labels? Can we somehow featureize this image and then enable a series of downstream genomic analysis associating this embedding with RNA-seq genetics and other data layers that we have in this data. And so this is different from what we did before, because now we are really just featureizing the image to enable statistical modeling of images. And so I think we ended up using SimClear, which is a quite simple framework for contrastive learning of visual representation. And I thought this is a good motivation also to tell you a bit about this methodology, and discuss a bit of the technical details here. As sort of a mini tutorial you can think within this talk. So the idea here is to learn representations of images that are invariant augmentations. So the model is optimized to learn embeddings of augmentations of different images that are similar, and while pushing away embeddings of different images. And this is done by optimizing this loss, this normalized temperature scaled cross entropy loss, NT-cent. And for each tile, basically you want to maximize the similarity with its augmentation while making it different from all other embeddings. And the representation that we learn, these objects here after the convolutional neural network, are evaluated by their ability to predict labels in a multi-class logistic regression. So this is not our work. We applied this. This was from Google Brain, came out a few years ago, and this really transformed the way we think about unsupervised learning. The main application there was ImageNet, which is a quite tough supervised task, but what they showed is that if extract features using this procedure, you can get predictions based only a single multitask linear regression. You can get predictions that are in line with the fully end-to-end supervised approach, which is quite astonishing.
So can you maybe spend a little bit more detail explaining this? So you're looking for embeddings, right? That if you, and then you run a multi-class logistic regression, and then, but of course, like the system doesn't know
the labels.
The
yep.
labels. So you're basically trying to you run the multi-class logistic regression on everything, and then you want to attract the images that have similar predictions and repel the ones that have more different predictions?
So this is done only for featureization. So you can think about this first step. This is totally unsupervised. The labels are not involved. It's just done for featureization of the images. And the way this visualization is trained is in a way that embeddings of different augmentations of the same image attract each other, they are similar, while embeddings of augmentations of different images, they're different.
So that has nothing to do with the multi-class regression?
The multi-class logistic regression is just how the quality of this visualization is evaluated a posteriori. Later.
What is NLP then?
Multi-layer perceptron,
Oh,
and that's just to go from this higher dimensional to lower dimensional on which then the loss is computed.
I see. Okay.
And
Logistic regression is in the higher dimension afterwards?
that's a good question. The logistic regression is done here, and they show that doing it here is better than doing it here. You know, it's a good question because where you do it is not clear, and they tested this in the paper.
Because the multi-layer perceptron is like just an enhanced version of logistic regression. So
Yeah, but the loss is not supervised, right? Yeah.
But it's not clear why. Okay, logistic regression is just a single hidden neuron. So MLP with a single hidden neuron. So okay.
Yeah, I think it
It
has.
seems like it could be included in this architecture.
No, but the idea is not to. The idea is not to, because you want to featureize your images in a way that is independent of the label. This is just learning a featureization that preserves most semantic information in the image.
Right, but you want to do well in this task, so maybe it would make sense to use both those functions somehow in learning.
Yes, the point there is that for many datasets you can have small datasets actually having a label. You know, if you semi-supervised learning task, that would perform very well. Another application that's the one that we consider is where, for example, we just want to do a genetic analysis of an image, and we don't know where to start. And so we use these methods to featureize the image in the first place, and then we can do standard statistical modeling, GWAS, and so on and so forth. Without the logistic regressions. Yeah, it depends. Like we do association testing, with somehow related. Okay, so a few technical details. So just a different representation, the same model. So given an image, the authors propose considering two different augmentations to get the transformed image, and then pushing this through a convolutional neural net to get the first embedding, then a multivariate perceptron to get the second embedding, and then they have this similarity that maximizes agreement between different augmentations of the same image. And the first question is which augmentations, and they test different combinations, and they found that the composition of cropping and color jittering really stands out. So basically, the fact that the model tries to make this type of augmentation having the same embeddings, and you can see these are very diverse, really empowers these embeddings in terms of content. Another insight from this paper is that this type of training procedure, unsupervised training procedure, benefits more from larger networks compared to supervised models. And that sort of makes sense because this tries to keep most of the information from the image independent on the label, not independent, but not being guided from the label. And so here you can see the top one accuracy on the ImageNet dataset versus the number of parameters in millions of the neural net that's used. And the red stars are, they correspond to a ResNet 50 trained for 1,000 epochs. And you can see that considering a higher number of channels in the network, so increasing the number of parameters, you get much better compared to the improvement in the supervised model. And if you now look at the biggest capacity models, these performances are pretty similar. But the red star did not use the labels for featurizing the images. And then finally, maybe the last bit that I think it's important, that they find that in training of very large batch sizes is highly beneficial. And so you need then to use optimizers to work well with large batch size training, which may be a challenge in some settings. But yeah, this was important because when optimizing this type of loss, it's important to have many negative samples. So you want to you have positive samples would be different augmentations of the same image, but then you want to make each image being different from all other images, and having many negative samples is important to get good representations. And all the evaluations that are done from these models are based on these multitask logistic regression a posteriori on the labels. Great. So how we used it? So we use it for featurizing our tiles that I was showing you before. So we learn representation that are invariant to rotations, flips, random cropping, color jittering, and blurring. Blurring was specific to these type of images that sometimes it happens as an imaging artifact. So it's also nice to add some of the imaging artifacts that we know occur in H&E when training these models. And then that's not a deal, but what we did to get biopsy level embedding, we just averaged. So remember that these tiles, they are just more parts of a bigger image. And ultimately, we want to featureize the whole image. So we did something quite simplistic here. We just averaged the embeddings of tiles from the same biopsy to get biopsy level embeddings. And yeah, I'll have a slide on how we're studying these now. Then again, the question, can we learn quantitative embeddings on liver histology beyond disease labels? Yes, for example, using this procedure. And now we want to compare the prediction accuracy that we get from this machine learning embedding versus the pathology scores that we were working with before, and also the machine learning version, the machine learning enhanced pathology scores that we derived before. These are the dimensions in parentheses, so these are, of course, the much higher dimension. And yes, so the first question is, now that we have these features, can we predict the pathologist label just from this featureization of the biopsies? And the answer is yes. We actually, of course, we are worse than the supervised model, but the results that we get are still quite meaningful. If we look at the top tiles, we can see very good agreement with the features once again. But then the benefits, when we then do genomic analysis and associate these embeddings with the gene expression, we see lots of genes being associated, being predictable from these embeddings rather than the supervised scores that we derived before. Great, last bit, and then we can wrap it up. A thing that we are very interested in is in detecting effects of genetic variants in tissue. This enables us to look at how disease risk loci may impact tissue function. And we did this focusing on variants that have been associated with liver dysfunction. And we were able to predict using a linear mixed model from this feature the genotype at PNPLA3, which is a risk variant for NASH, fibrosis, and steatosis. So these are out-of-sample predictions that we get. And this variant was not associated with the pathology scores. We could only see it when we got these unsupervised visualization of images, meaning that these changes that do not necessarily correlate in with disease progression in this cohort, but we could identify a histological phenotype associated with the microdroplets within the hepatocytes, which we found quite interesting and in line with what we know about PNPLA3. And now I told you that we did a quite simple assumption when considering just the average of embeddings from different tiles in biopsy level embeddings. Now we are extending this type of models, bringing in these principles of attention. So how should we aggregate tiles from, how should we aggregate embeddings from tiles of the biopsy using these attention mechanisms? And the challenge here is that since usually we want to do genetic association studies, we want disaggregation to be really fast. And so, yeah, that's where the computational challenge is, and I'm not going to go into details. You should talk to Shubham, who's here in the audience, who's working on that. Great, so this concludes this second part. Self-supervised approaches can extract general histological features only based on principle of invariance and variation in the population. These features enable genomic analysis of histological traits, and this really enables us to make